Okay, today we are going to discuss about the kingdom fungi. Let's start. First, we'll discuss about its general character. Normally, fungi are achlorophylous in their habitat, means they don't have no chlorophyll present. They don't have any chlorophyll present. So, as they don't have any chlorophyll, so they are unable to prepare their own food. So, they are heterotrophic in their mode of nutrition. And they are normally eukaryotic organism, means the cell do contains nucleus and membrane bound organelles. So, they are eukaryotic. And these organisms, these fungi, they produce different types of spores. And they produce different types of spores produced by this fungi. And the body is thalloid organism. What does thalloid means? Means the body is not differentiated. The body is not differentiated. The body is not differentiated into stem, leaf, and roots. If any organism where the plant body is not differentiated into stem, leaf and root, then they are called as thalloid. So normally they don't have stem, they don't have leaf, they don't have root. So we do call them as thalloid organisms. Got it clear? Now let's move. These fungi includes diversified organism, means they contains variety of organism ranging from unicellular yeast, there is unicellular yeast, to a complex organism that is mushroom, which is a complex organism. When we'll discuss about mushroom, so mushroom is a complex organism. So from unicellular yeast to a complex edible organism that is called as mushroom. The non-edible toadstool is one kind of fungi, is the best example of complex organism with a diversified habitat. Now, normally they are cosmopolitan, means they are present everywhere in the world. They are found present in air, they are found water, soil, and also they are present on animals and also on plants, thereby causing diseases. They are mostly found in warm and humid areas, so where moisture is present. If moisture present and the condition is warm, then the growth of fungi is maximum. And they have great diversified morphology. They have a variety of morphology with variety of habitats. Means, okay. Now let's go for a question. Why do we normally keep the food in the refrigerator? Why? Because the refrigerator protects a food from bacteria and other fungal organisms, because normally fungi and other organisms, they are unable to grow at low temperature. They become inactive, because we now discuss the fungi they need warm temperature and moisture, but in cold storage in -degree centigrade we'll find that they are inactive. So for which our food gets protected from this organism if we keep it low temperature. As we have discussed, they are cosmopolitan. It is found in air, water, soil, animals, plants, and they are mostly terrestrials, means they are mostly found on land. They are mostly found on land, so they are mostly terrestrial in the habitat. And they prefer warm and moist places to grow. They may grow on tree bark, they may grow on cow dung, they may grow on wood, they may grow on burnt wood, or they may grow on keratinous material, that is hair or any keratin proteins. They can grow everywhere. They also grow on dead cellulosic materials. If a plant body is dead, and the dead wood or the dead cellulosic material, the fungi are able to grow on them. For examples, they grow on crop residues, they grow on cow dung, they grow on wood, so called as epigylic, they grow on burnt wood, so called as xylophilous, and they grow on keratin materials proteins, that is called as keratinophilous, means they grow on hairs. And the body is haploid. Normally the body is N, it is the haploid mode of thalloid. What is thalloid? We already discussed. And it is not differentiated. The thalloid means it is not differentiated root, stem and leaves. So they are called as thalloid. And they are multicellular except yeast. Yeast is unicellular. And Syncytium is also unicellular. Both of them are unicellular. So except yeast and Syncytium, all other fungi are multicellular eukaryotic. Only yeast and Syncytium, it is unicellular and eukaryotic. And normally the fungal body is made of a thread-like structure. The fungal body is made of a thread-like structure, and this thread-like structure is called as hypha. So normally the body of fungi is called as what? Hypha, because it's looking like a thread-like structure. So this is the fungal structure, let's discuss. And here what we find that this part, these are the cell present, these are the cell unicellular present. Inside the cell we find the nucleus are present, and these are called as the hypha. These are called as the hypha, and this hypha interwoven group of hypha together is called as mycelium. It is called as the mycelium if they are present in interlocking arrangement, and in between them we find a division line present, that is called as the septum. So this is the hypha present. The single thread-like structure is called as the hypha. But if the hypha is interwoven, if the hypha is networked, then we do call them as mycelium. Got it? So this is about the structure of the fungi. Now let's move that the arrangement, if the arrangement is in a crisscross manner, if there is a network-like manner, so we do call them as mycelium. So mycelium is the group of hypha present in network form. So this is the structure of the fungi, but the actual structure is called as the hypha, and each structure is called as hypha. Each thread is called as hypha, and the group of thread is present is called as mycelium. The group of thread present together is called as what? The mycelium. Let's move. And normally the hypha may be aseptate, multinucleated, or which is called a coenocytic. Means suppose this is the hypha present, and if the hypha don't have septum, then we find what? The nucleus is multinucleated. Got it? So this type of hypha is called a coenocytic hypha, and here no septum is present. As no septum is present, it is called as aseptate. Got it? As no septum is present, is aseptate, and we find multinucleated. The other name of multinucleated fungi is called as what? Coenocytic. So this type of hypha is called as multinucleated hypha, or you can call them as coenocytic hypha. Now most of the fungi, the mycelium is septate. Normally some fungi aseptate, but most of the fungi we find the mycelium it is septate. Means it is uninucleated, means they have the septum present. It is the septum present here, and these cells are uninucleated. Uninucleated. Got it? Multicellular, no, it is many cell present, multicellular, but due to the presence septate is called as septate hypha. And the septum is not complete, but it may contain certain pores are present. So we find that in between the septums, the septum is not complete, and they contain small openings present, and these openings present in the septums are called as septal pore. These are called as septal pore, and these septal pores do help in exchange of materials between the cells. This is the septum. So septum is not continuous, they contains pores in between them that do help in exchange of materials within the cells. Got it clear? Now let's move. So here we find this is the coenocytic hypha and this is the septate hypha. These are two different types of hypha where we find in lower fungi, mostly in lower fungi we find coenocytic hypha, but in higher fungi we find septate hypha. We'll discuss further. The septum may have simple central pore as in Ascomycetes. So normally in Ascomycetes what happen? It is the highermost fungi. We find the septum contains septal pores. So these are called as the septal pore. And these septal pores are present in the group that is called as Ascomycetes, and these are called as the septum. And Ascomycetes is normal, the advanced fungi. It is the advanced most of fungi. But in higher fungi like Basidiomycetes, the septum is present, but it is in the doliiform form. For example, hypha present. Here whatever, the septum is present in a hook-like manner. So here what we find, the septum is present like a hook-like manner. Got it? So this type of hook is called as doliiform septum. This is called doliiform septum. Got it? And this is present mostly in Basidiomycetes, and this is the septum part. Got it? So in Basidiomycetes we don't have central pore, because in Ascomycetes we found central pores are present. But in Basidiomycetes we find a modified pore present at the septum, and this type of modified pore present is called as the doliiform septum. And it has pore through which nucleus can pass through. Yes, through these pores the nucleus can move here and there. So here we find the central pore present. So normally what happen? In this particular diagram, we find what? the central pore is present. This is the central pore. This one is the central pore. This is the central pore is present in between. This is central pore present. And this is the structure of hypha. And here hypha we find, this is a single cell. We find the nucleus present. We find the mitochondria is present. We find the ribosomes are present. We find the vesicles are present. Here the plasma membrane present. The cell wall is present. And also we find Golgi bodies are present, and the Golgi bodies endoplasmic reticulums are present. The Golgi bodies present in single cisternae form, unicisternae. This is the unicisternae form. We'll discuss later everything. Okay, now let's move. The pore possesses a barrel-shaped swelling called the parenthesome. As we find, here is the septum. This is the barrel-shaped swelling. Got it? So this swelling is called as the barrel-shaped swelling. So this barrel-shaped swelling is called as parenthesome, and which we find in Dolifer septum, and normally it is present in Basidiomycetes. Now in Septate mycelium, individual cells may contain single nucleus or an intermediate phase of two nucleus. Sometimes what happen in septum phase, any septum, suppose any septum, either we may contain single nucleus, or sometimes we find two nucleus present. And this two nucleus present is called as dikaryophase, dikaryotic nucleus. And this dikaryotic nucleus is present as an intermediate phase, we'll discuss later. But normally in septal mycelium we find a single nucleus is present, single, this is the nucleus present. But in interphase we find certain dikaryotic nucleus present where two nucleus are present of different strain or from different hypha. We'll discuss later. So this is what about the septal nature of this particular hypha. So that is called as the dikaryotic feature, and this is called the secondary mycelium. Monokaryotic is the primary mycelium, because in monokaryotic we find single nucleus present. This is called as monokaryotic mycelium. It is monokaryotic, and in monokaryotic it is called as primary mycelium. This monokaryotic is called as primary mycelium. Got it? But when we move to secondary mycelium, we find what? Sometimes we find two nucleus are present in the interphase phase, and this two nucleus stage is called as dikaryotic phase, and this mycelium is called as the secondary mycelium. Got it? So this is the distinct feature, advanced feature of most higher fungi. So here you find is the Doliper septum. This is septum present here, and this is the Doliper septum present, and we find the swollen rims are present here. Parenthesome, these are swollen, but the swollen rims are present, that is called as the parenthesome, and this is the modified form of the Doliper septum present in Basidiomycetes. The cell wall of hypha is made up of chitin or cellulose, but mostly this is a chitin, and this is called as fungal cellulose. This is called as fungal cellulose. So it's the distinct character of all fungi. And in some fungi the cell wall is found of cellulose, and mostly cell wall made up of cellulose found in Oomycetes. In Oomycetes fungi we find the cell wall is made of cellulose, whereas in higher fungi we find the cell wall is mostly made up of chitin, and Oomycetes is the lower fungi, got it? Or it is called as the algal fungi. It is the algal fungi, got it? So it is the algal fungi. So normally in some this algal fungi we find cellulose is present in its cell wall, but most of the fungi, that is higher fungi, that contains chitin, and chitin is the parent material of this fungal cell wall. Reserve food materials is oil and glycogen. Reserve food materials is oil and glycogen, and we have also studied that glycogen is also reserve food materials of animal. It is a reserve food materials of animals. So most of the animals also contains the reserve food materials as glycogen, as we already discussed. So in fungi, oil and glycogen as the reserve food materials. And we normally discuss that when in fungi we find nucleus present, but unicisternae means single cisternae is the present. This is the special character of fungi, means single cisternae is present. Golgi body contains stacks of cisternae. This is called a stack of cisternae. These are called as cisternae. So most of the animal we find 4 cisternae present. In plants we find 20 cisternae present. So each are called as the cisternae or saccules. But in fungi only 1 cisternae present. not stack of cisternae present. Mitosis in fungi is karyochoresis. So what does this means? So mitosis in fungi is a different type of mitosis in somatic cells where we find it is karyochoresis. What is karyochoresis? So normally what happened during mitosis, within the nucleus the spindle fibers are present within the nucleus. So nuclear membrane is present and also the spindle fibers are also found to be present. So this is the nature of called as choreosis. So this is the nature is called as what? Choreosis. Let's move means they contains intercellular spindle formations. Means normally they have intracellular spindle formation. And the nutrition is mostly saprophytic, parasitic and symbiotic. Saprophytic means the mostly depend upon dead, parasitic means living host and symbiotic means mutual. As we find in lichen. In lichen we find algae and fungi are present in mutual benefit symbiotic. So they are mostly the unicistern and Golgi body present. The mitosis is present special type where the intranuclear spindle formation is present, means within the nuclear membrane. Nuclear membrane is distinctly present. Within it we find spindle formations are found, that is called as karyochoresis. And the mode of nutrition is heterotrophic and it is three types: saprophytic, parasitic and symbiotic. So this is the fungal cell where we find these are the Golgi body cisternae are present, single cisternae. These are the present nucleus present, inside nucleus present. These are the endoplasmic reticulums are present. We find the vacuoles are present. In some, not in some, we find the vacuoles are present. And certain vesicles are also found to be present. This is all about, this is the parent body present. The bigger one is the parent body present. And this part is the bud. We'll study about the bud in yeast, the bud part, and this is the parent part. This is the parent part present, and in the parent part we find this is the nucleus present, the chromatins are present, within the nucleus are present, endoplasmic reticulums are also present, Golgi bodies are present, mitochondria present, ribosomes present, peroxisomes present, peroxisomes present. Got it? And after the bud is detached from the parent, we find a demarcation is present here, and this is called as the bud scar. This is called as the bud scar. After, when the bud detached from the parent, then we find a bud scar. And here the septum is found to be also present. So this is about the eukaryotic cell structure of fungi. Now let's move that this particular fungi, in most of the fungi, they contain two different phases. One is the vegetative phase, yes. Another one is the reproductive phase. Got it? So in a single fungi, we find two phases are present. One cycle completes vegetative phase, another cycle completes the reproductive phase. So the two phases are present to complete the life cycle of fungi. But when we go into the vegetative structure, the vegetative structure is very microscopic and hidden, and it is unable to visibly naked eye. So these vegetative structure are mostly present hidden within the substratum, but the reproductive structure can be visible because when the spores comes out, when the conidia comes out, when the sporangial spores comes out, that can be visible with the naked eye or with the microscope easily. The fungus reproductive phase comes after maturity of the vegetative phase, as we have discussed. But unicellular yeast, in yeast a modification is there. What? Now the single cell of yeast will behave as vegetative phase, also behave as a reproductive phase. So they don't have a separate phase. But in other multicellular fungi we find they have a separate phase called as vegetative phase, and they converge into another phase where the fruiting body comes out, where the spores are comes out, and is the reproductive phase. Got it? So in a higher fungi we find two different phases which are distinct. We can differentiate that this is vegetative phase, they don't have fruiting bodies. These are the reproductive phase where the fruiting bodies are found to be present. But whereas in yeast, we don't find. Single cell yeast will behave as vegetative phase. When it becomes mature, then they go for reproduction. That is budding. Got it? So yeast is exceptional. So this nature of yeast is called as holocarpic. So this nature of yeast is called as holocarpic. If a fungal body behaves both reproductive phase and vegetative phase simultaneously, then that fungal body is called as holocarpic, and the best example is yeast. So this is the structure of yeast. This is the eukaryotic yeast, and this is the budding part, the budding structure. This is the bud part. Got it? So here what we find, now this same vegetative phase, when it gets mature, gives out bud, behaves as a reproductive phase. There is no different phase. In a single phase we find, in a single structure we find vegetative phase as well as reproductive phase. So this is called as holocarpic. So this nature is called as what? Now holocarpic. Got it? But in other fungi we call it as eucarpic, because we find a differentiated vegetative phase and reproductive phase. So differentiated vegetative phase and reproductive phase is called as eucarpic in nature. Okay, now let's discuss about the reproduction of fungi. So fungi can reproduce in three modes: vegetative phase, asexual reproduction, and sexual reproduction. Three nature mode of reproduction is present in fungi. So vegetative reproduction occurs by the following method, that is mostly by fragmentation. Some of the fungi, they undergo fragmentation, means the hypha breaks into small pieces. And these pieces are called as fragments, and these fragments then develops into new mycelium. So this method of reproduction is called as vegetative phase. And here what happens, some part of the mycelium or hypha gets decayed or is mechanically injured, gets into fragment structure, and these each fragments then develops into new individuals. So each fragment then develops into an independent mycelium, and this nature is called as vegetative mode of reproductions. And in some fungi we find fission present, where we find the cell divides, the cell division takes place, and the cell divides into two cells, and this is called as fission. So one single cell by division gives us to two cell, and this type of simple division is called as binary fission or by fission method. In some fungi we find fission is also possible. Budding is mostly present in the fungi yeast, where the parent structure gives out the bud. So this is the bud and this is the parent. So when the favorable condition comes, the bud gets detached from the parents, and then genus then develops into a new vegetative mycelium. So the bud cuts up from the parent cell and develops into a new individuals, as we have discussed. Asexual reproduction is mostly by spores. So vegetative reproduction is mostly by fragmentation, fission, and budding. We have discussed. Now we will discuss about asexual reproduction, and this asexual reproduction is mostly by means of spores. So let's discuss about asexual reproduction. So spores are single cell specialized structure for reproduction. So spores are single celled. These are single cell reproductive structure, specialized structure. Why specialized? Then they are only meant for reproduction, to maintain the generation, to give a new individuals after the germinations. So spores get dispersed and germinate to produce a new mycelium. For example, suppose this is the structure present. For example, suppose I am drawing, this is the sporangia present, for example. And inside sporangia we find the spores are present. Got it? And the stalk is called as sporangiospore. Sporangiophore. The stalk is called as sporangiospore. So normally what happens when the sporangia gets matured, what happens? The sporangia is broken down and all the spores are dispersed out. So all the spores gets matured and dispersed out, and these are called as the spores. Got it? And these spores are haploid, these are N. They germinate to give rise to a new mycelium or hypha, and this hypha then converts into mycelium. Got it? So after germination, each spore develops into a new mycelium and continue the life cycles. So this is what about the asexual mode of germination, means spores gets dispersed, germinates to a new mycelium. So the spore produce asexual mode of reproduction, and they are normally by mitotic division, because we know very well the plant body is itself is haploid. So the spore is also haploid. So if plant body is haploid, spore is haploid, so definitely mitosis takes place, and through mitosis the spore is produced, for which we do call them as mitospores. So mitospores are the special type of spores produced by asexual reproduction by the mode of division that is called as mitotic division. Got it? So this is, let's talk. This is what we are discussing about the spore. Now let's see. So these are the sporangia. This is the sporangia present. This is the sporangia present. And we have entered into the sporangia because fruiting bodies are the sporangia. Inside sporangia, we find this structure, the spores are present. And this stalk is called as this sporangiospore. This is called as sporangiophore. Phore means stalk. Phore means stalk. Got it? And this is a total mycelium present, and these are the septum present in between them. So when condition comes, the sporangia breaks out and the spores get liberated and develops into a new mycelium. So, now let's move. In some fungi, those who are aquatic, they behave zoospore, animal-like nature, means these spores contains flagella. So if any spores having flagella, then these are called as zoospore. Got it? And zoospore are mostly for aquatic organisms. So aquatic members of fungi contains the movement like spores. These spores can move. They are motile. So these are motile spores. So normally all motile spores are called as what? Juju means animal-like behavior. So zoospore. So all motile spores are called as what? Now zoospore. So zoospore may be uniflagellated, may be biflagellate. Either zoospore contains one flagella or zoospore contains two flagella, either present at the anterior end or they are present at the lateral end. Kidney cell lateral saprolegnia contains this lateral arrangement of flagella. So zoospores are naked unilateral structures called as zoospore, formed from zoosporangia. Let's move. So this is the zoosporangia present. Normally this is the zoosporangia, this structure the zoosporangia is present. Within those zoosporangia, these zoospores are produced. Got it? So this is a germination tube. When the germination tube develops, this zoospore breaks out and gives out these zoospores. And these zoospores are of different types. Got it? And they have flagella present. These are the flagella present. These are the flagellas are found to be present on this particular zoospore. Now let's move. They germinates to give rise to new mycelium, as we have discussed that any spores which germinates, they will give rise to new mycelium. And biflagellated zoospore is again classified into two types, into two kinds, biflagellated. So what are they? Let's move. In Saprolegnia, this is a species, this is Oomycetes. Saprolegnia is a lower fungi, Oomycetes. We find pear-shaped or pyriform zoospore with two flagella present at the lateral end. Two flagella present lateral end. If this nature is found, it is called as pear-shaped. It is called as pear-shaped or pyriform zoospore. Got it? But in some saprolegnia, in the second stage of saprolegnia, we find two different types of flagella present at the lateral end. The kidney-shaped structure or bean-shaped, and we find this is the secondary zoospore present, but primary zoospore is the pyriform nature. This is the pear-shaped, that is the primary zoospore. In saprolegnia, we find two types of life cycle, primary mycelium, secondary mycelium. So we find primary zoospore followed by secondary zoospore. So the secondary zoospore, what we find, the lateral end the zoospores are present. These are biflagellated. Got it clear? And this biflagellated nature, having two zoospore in the life cycle, is called as diplanetism. The phenomenon of having two types of zoospore, and the best example is saprolegnia. So saprolegnia is the best example of diplanetism. So here this is the zoosporangia present, and this is the, we'll discuss later about these different types. This is a zoosporangia present here, and here the zoospores are liberated. This is a single cell flagella zoospore, and this is the biflagellated zoospore present. And this one is the conidia. We'll discuss about conidia. These are the conidia, oidea square like structure present. These are the conidia present, and these are the sporangia present, and inside we find the spores are present. Got it? So these all are the asexual mode of reproduction. One is the zoospore, another one is the sporangia spore, or this is called a sporangia spores are present. One is the conidia present. These are the conidia present. We find the conidiophore present, and this conidiophore gives out branches called as rami. From the rami we get small branches present, that is called as medullae. From medullae different branches present, that is called sterigmata. From sterigmata we find the spores present, which is called as conidia, arranged in chain like manner. And here these are called as the chlamydospores, wherein chlamydospores inside the hypha cell develops into spore. This hypha cell develops into spore called as chlamydospore. And this one is called as the oidea, where we find oidea. What happens whenever the sugary medium is present, when maximum sugar is present, then at that time each part of this particular hypha, they liberate out into, they detach into small spore like structure. Here what happens, chain like spores are present called as coronae. Here what happens, any spore they'll simply liberate out. So this is the condition of oidea. They all are of asexual mode of reproduction. Okay, we'll discuss later. Now let's move. Sporangia spores are thin non-motile. Spores produce endogenously. Yes. Now we have discussed. This is sporangia. This is sporangia. Inside sporangia, we find the spores are present, and these are called as the sporangiospore or called as spore. And the stalk present is called as the sporangiophore. Sporangiophore. The stalk is called as sporangiophore. And normally what happens, it is present within the sac. So there are endogenous spore. So within sporangia, the sac, within the sac, the sporangia is present. Got it clear? Now, during favorable conditions, these sporangia liberates and gives rise to new mycelium, which we have discussed earlier. In the best example of sporangia, spores are found to be present in mucor and rhizophos. So rhizophos and mucus undergo reproduction by means of special spore that is called as sporangiospore. Conidia. Conidia, just now we have discussed that this is the structure present, and from structure we find the branches are present. From the branches we find the chain-like structures are present. These chain-like spores are present. So these chain-like spores present is called as conidia. Conidia. So conidia contains chain-like, which is the best example of penicillium. We'll discuss about. And the stalk present is called as conidiophore. Got it clear? So the chain of spores present is called as conidia, and this is exogenous because it is present outside the sac. It is exposed out. So these are exogenous spores. Clear? They are arranged, yes, they are arranged in chains on conidiophore. The conidiophore is the stalk, and the best example of conidia is aspergillus and penicillium. These are the best example of conidia mode of reproduction. This is sporangiospore, we have discussed earlier, and this is conidia. This is conidia, got it? And these are the chains present, and these are the conidiophore. This the all the conidiophore, but this is conidiophore. This is ramping, and this is steric medulla, and steric. These are the spore present. These are the conidia present, which liberate to give rise to new individuals. And here what happened? Now each chain liberates out. These are liberates out. Here what happened? They are present in stalk when the terminal one gets matured, then liberates. Here simultaneously one by one gets matured out, and each individuals are called as oidea, and is mostly present in sugary medium objects. Got it clear? Now let's move. In some fungi under unfavorable conditions, we find thick walled resting spores are present to withstand unfavorable conditions. And these spores later get separated from each other and develops into new. And they may be terminally present and they may intercalary present, and they are called as chlamydospores. They may remain viable for several years. Yes, they can remain viable for several years. On return of favorable conditions, these spores germinates into new individuals, and thus these are called as chlamydospores, and that helps in perinations. So just we discussed that certain spores are present, means this hypha, endogenously this hypha develops into spores. So this hypha develops into spores like structure. So these spores so form within the hypha is called as chlamydospores. And on the adverse conditions they undergo, that is dormant. And when conditions are favorable, they germinate. So they help in perennation nature. Rhizophos is the best example of chlamydospore formation. Rhizophos. This is the best example, very important exam point of view. Non-motile thick-walled spores also develop in sugary-rich medium, and I have just discussed these are called as oidea. On sugary-rich medium, we find non-motile spores are present. These are called as oidea. And in yeast, we have already discussed the budding stage is present. These are the bud present. In yeast, we find the buds are present. Oidea are also called as torula. This oidea is also called as tubular stage. Very important exam point of view. Now let's move. We have discussed vegetative reproduction. We have discussed asexual reproduction. Now we are going to discuss about sexual reproduction. In sexual reproduction, we find oospore given by Oomycetes, ascospores given by Ascomycetes, basidospores given by Basidomycetes. All these spores are called as sexual spore. So we have already discussed asexual spores, but normally sexual reproduction don't have spores because they do have gametes. But in fungi it is different. Fungi gives rise to sexual spores in the form of oospore by Oomycetes, ascospores by Ascomycetes, and basidospores by Basidomycetes. Let's discuss one by one. So the various spores are produced in special structure called as the fruiting bodies. The fruiting bodies are called as ascocarp for Ascomycetes, basidocarp for Basidomycetes, which contains asci, the sac, and basidia are also the structure that do liberate the exogenous spores. Let's discuss one by one. In ascospores, we find non-motile spores are present in a sac-like structure. Suppose it's a sac. The sac is called as asci. Within the asci, we find 8 spores are present. So within the asci, we find 8 spores are present, and these 8 spores are called as ascospore. And these ascospores are present with this special sac-like structure asci, and the asci are found on the fruiting bodies, and this is called as the fruiting bodies of Ascomycetes. Got it? But talking about, yes, 8 ascospores are produced, 8 ascospores are produced. Yes, this is the ascospores present, 8 ascospores are present, and the singular is, this is called as the asci present. This is a structure of Ascomycetes, ascospore, sexual spore. But in Basidomycetes, what happens? Now, normally what happens? They have fruiting bodies like structure called as basidium. Suppose this is basidium present. This basidium gives us 4 steric and from this 4 steric the spores are produced, and these are exogenous. Ascomycetes is endogenous, and these are called as what? Basidiospores. Basidiospores. Got it? These are called as basidiospores, and this structure is called as basidium. So this structure is called as basidium. Got it? So Basidomycetes, we find basidium present, and outside basidium we find 4 outgrowths are present, and these outgrowths gives out the structure that is called as 4 basidiospores are present. So 8 ascospores present endogenously, and 4 basidospores are present exogenously. Got it? So here we got the structure. Let's discuss. So the first one is called as the ascospore. So this is the ascus present, the sac is present, ascus, and inside the ascus we find the ascospores are present, which is 8 in number, within the sac. So these are endogenous. So they are endogenous. Here what happened? The fruiting body basidium, and they contain the stalk that is called steric. From the stalk we find exogenous basidiospores are present. So these are 2 different structures of Ascomycetes sexual spores and Basidomycetes sexual spores, and their fruiting bodies. Now let's discuss. Sexual cycle involves 3 steps. What are they? Plasmogamy, karyogamy, and meiosis. Plasmogamy, which means the union of protoplasm. So this is the cell present, this is the cell present. So initially when both the cell comes, the union of protoplasm takes place, and this is called as plasmogamy. The first stage is union of protoplasm. After that contains what? Now both the nucleus then fuse, then it's called as karyogamy. Got it? But as we have discussed that the plant body itself is haploid, but after karyogamy it is diploid, so they must return back to the plant body. So to return back to the plant body, they should undergo meiosis. So 3 stages are present. One is plasmogamy, karyogamy, and meiosis. Got it? So sexual reproduction is involved in union of protoplasm, union of nucleus, and again meiosis of nucleus, reductional division of meiosis to develop into a parent body. Now let's move into one by one. First we'll discuss about different types of plasmogamy. First one is called as planogametic copulation. So here the gametes undergo union, and plano means they are having the power of movement, Aplano means they are non-motile. Plano means they are motile gametes. Let's move. And the process involves two gametes of opposite sex to get union. And the fusion takes place with the motile. If it is motile, then it is called as plano. So plano stands for motile. Aplano stands for non-motile. So here what happens? The fusion between plano-gametic copulation, matlab fusion between motile gametes do takes place. So it is of three types. One is called as isogamy, where union of same type of gametes takes place and both are flagella. This is called as isogamy. Anisogamy, where we find union takes place between different types of gametes, different size of gametes. Oogamy. Oogamy, one is non-motile, where the female is non-motile. So between non-motile and motile, and the sperms are motile, the male are motile. So between motile and non-motile, it is called as oogamy. Between different sizes, it is called as anisogamy. Between same size, it is called isogamy. And this three type of copulation is called as what? The first type of protoplasmic union is called as what? The planogametic copulation. So here we have already discussed this one. Now let's move. In the result, after planogametic, what we find, the zygote is produced, diploid zygote. Got it? Oomacetes show planogametic copulation. Got it? Which organ? This is the main question. Which organism show planogametic copulation? Oomacetes. Oomacetes are called as the lower fungi or the algal fungi. Lower fungi are called as the algal fungi or cellulosic fungi. So during the process of two gamete union, one contact with the other. When two gametangia contact, for example, suppose this is the male one present and suppose this is the female one present. So these are called as gametangia. Each sex organs are called as gametangia. So gametangia is the sex organ. So when two gametangia contact, now this type is called as gametangial contact. The fertilization developed is called as gametangial contact. Got it? So when two male and female gametes contact and undergo fusion of protoplasm, and that type of formation is called as what? Gametangial contact. Normally gametal contact, what happens? Suppose this is the female one is present, oogonium present, and suppose here the male antheridium is present. The male will produce a fertilization tube, and through this fertilization tube it will discharge the sperm, a spermatozoa. So this is the fertilization tube. So this type of fertilization, this type of union of protoplasm is called as gametangial contact, and it is by Pythium and albigio are the best example. Here this one, this is the gametangial contact. Got it? How it takes place? Got it clear? Now let's move. In the gametangial contact, what happens? The entire content of the gametangia is move into it and get involved. So the result in the formation of two gametangia, they fuse together. Yes, as the result, these two gametangia fuses to form a single, which is 2n. Then they undergo karyogamy. After karyogamy, then they undergo meiosis to produce a sperm. And the best example is mucor and rhizopus. Rhizopus and mucor as the best example of gametangial copulations. So here we find gametangial copulation. In gametangial copulation, what happens? Now, normally in gametangial copulation, we find what? Now this is a plastid, is a method, two different hypha. It is one kind of hypha, it is another kind of hypha. So these two hypha will come to each other and the terminal part will swell and they get fused. So after fuse, what happens? So after fuse, so they both are gametes fuse. From the fusion, they gives out these fruiting bodies. Got it? This is how. And from this fruiting body, the spores being liberated. This is also after fusion, this is a fusion, and from this fusion, the fruiting bodies are developed. From the fusion, the fruiting bodies are developed. And these fruiting bodies gives rise to the spores, liberated spores, and these liberated spores this comes out and gives rise to new individuals. Some fungi produce sperm-like structure, single cell sperm-like structure. These are called as spermatia. And these spermatia transfer through the agency of water or by wind or by some vectors like insects to a female hypha. So this is a type of hypha. It is a type of hypha, and these hypha produce small type of spermatia. These are spermatia. Spermatia is a type of hypha. They will come to another hypha, and this hypha is called as female hypha. They get attached on the female hypha and get fused. And this type of fusion is called as somatogamy. Somatogamy, because two hypha structure gets fused. So female is the receptive hypha and spermatia is the donor hypha. So as a result, this type of structure is called as what? The somatogamy. Yes, here the spermatia come receptive hypha, and after that the union and undergo somatic hypha. When both the hypha, for example, when both the hypha are fusing, means this is the vegetative structure, this is the vegetative, both vegetative structure are fused without any spores are produced, then this type is called somatogamy. And here sometimes spermatia produce, and this spermatia moves to the hypha receptive female, undergoes union, and this type is also called as what? Somatogamy. Different type of somatogamy is found to be present. Thus, diachroic condition is established. So Puccinia shows spermatization. Means in spermatization what happens? Suppose for example is the female hypha present. So female hypha is of single cell, uninucleated. Suppose for example this is the male hypha present. For example is the male hypha and this is the female hypha present. So this male hypha gives us these are the spermatia. The spermatia gets attached. So when the spermatia gets fused, what happens? The first one will bear two types of nucleus. One of male and one of female. And this condition is called as dikaryotic condition. Dikaryotic means two nucleus are present. And this is monokaryotic. This is monokaryotic. This is monokaryotic, but this is dikaryotic, where two nucleus are present. So this type of spermatization is mostly present in the fungi, that is called as Puccinia. Somatogamy takes place in most of the higher fungi. Yes, this is the basic character, is the advanced character of fungi. And formation of gamete is absent in such fungi. Where somatogamy takes place, then gametes are not produced. This is a very important point. In any fungi where somatogamy takes place, when the spermatia helps in reproduction, in that type of fungi we don't find any gametes are present. Your gametes are absent. So direct fusion of somatic hypha cells occurs due to dikaryotic phase. Just now we have discussed direct union. And in the agaricus, the mushroom, the somatogamy is the best example. So we should know in which fungi what type of reproduction takes place. After the union of protoplasm, we have discussed about gamete-angel contact, gamete-angel copulation, somatogamy, spermatization, planogametic copulation. We have discussed 5 types of planogamy. After planogamy, the nucleus takes place. So union of nucleus takes place. So here what are the protoplasm union takes place, and after that what? The nucleus takes place, and this union of nucleus is called as karyogamy. So what about this? We have just discussed dikaryotic phase. In dikaryotic phase, this present two different. After that, what happens is a clamp connection takes place. We'll discuss later in the Basidiomycetes, but still I'm telling you. Suppose this is the spermatia of one male and this is the female. So after mitosis, what happens? This male gets 2 and this gets 2. So one of this clamp connection is produced, and this clamp connection gives rise this one. This comes to this phase, this part. And what happens? This part becomes one part, and here we find this. So these are called dikaryotic phase. So we find dikaryotic phase like this present. We'll discuss later about dikaryotic phase in the Basidiomycetes part. In some fungi, the two haploid cells immediately union results in diploid cells where absence of dikaryotic phase. So if any fungi where the karyogamy is delayed, then dikaryotic phase takes place. But if karyogamy is not delayed, then directly it develops into diploid cells. So there is no intervening dikaryotic phase occurs. So such a condition is called as dikaryon. So such a condition is called as what? A dikaryon. In some fungi where we find the dikaryotic phase present called as dikaryon, But in some fungi, we find there is no diacritic phase present. Suddenly, we find the diploid cells are present after the union of the two nucleus. Now later, after parent nucleus fuses, that is the condition is called as karyogamy, and this karyogamy undergo fruiting bodies in which reduction division takes place, that is called as meiosis. So suppose this is the zygote present. The zygote is the karyogamy, and this zygote develops into hypha, which is meiosis, and from this they gives rise to fruiting bodies, and all are N. So to produce N, they undergo what? Now reduction division, that is called as meiosis. So leading meiosis occurs leading to the formation of haploid spores. Yes, meiosis takes place to produce haploid spores. So meiosis helps in formation of haploid spore. Kingdom fungi is then classified. Now let's move into the classification. So now mostly the fungi, we are talking about the general character of fungi. So kingdom fungi is classified on the basis of three conditions. One is the morphology of the mycelium. Second one is the mode of spore formations. The third one is the fruiting bodies. So fungi is classified on the basis of mycelium structure, on the basis of spore production, on the basis of fruiting body productions. So this is the fungi we have discussed, and this is the classification of fungi. So normally fungi is Phycomycetes. This Phycomycetes is called as the algal fungi. It is called as the algal fungi, or it is called as the lower fungi. It is called as the algal fungi, or we do call the lower fungi. So this part is of lower fungi, and this part is of higher fungi. This part is of higher fungi. Got it? So lower fungi is called as Phycomycetes or algal fungi, which is of two types, Oomycetes and Zygomycetes. Got it? But in higher fungi we find Ascomycetes, Basidiomycetes and Deuteromycetes. So in the next class we'll discuss details about Phycomycetes, Oomycetes, Zygomycetes and all other higher fungi. So this is the very important classification of fungi. So hope you all enjoyed, and thank you for watching my video.